Today, we'll just have a quick look into the ligand receptor analysis that we have on ScarfWeb. A little bit of a context to what example we're using today. This is a small patient sample dataset from a study on myocardial infarction. This is cells from one patient, but it contains two sort of groups. We have the normal cells and then we have the ischemic cells. They are combined here in one, but we are interested in comparing maybe the cell signaling in one. Maybe like what's the difference between cell-cell communication in the ischemic compared to just normal? For example, to keep things simple. So this is a convenient little analysis that you can use for discovery or validation into what sort of signaling mechanisms that might be happening. So it's a quick way to generate some candidates or potential candidates for hypothesis that you could probably test with further analysis or some other experiments you have in mind. All right, so quickly getting started here, we have the ligand receptor analysis. For primary grouping, we are choosing the cell type original. So these are the original cell type annotations made by the authors. And because we want to compare between ischemic and myogenic, we want to have sort of two different tables or two different runs. So we will first select, for example here, you can subset this data set since it's mixed for the ischemic, and then you can do another run for the myogenic, and then you can compare the two sort of results. And then here you can look into our documentations for more about the parameters you can use, but for now I will just leave them as the default ones. And make sure that you have the gene symbols, because the ligand receptor database, they rely on the gene symbols. And you can get started. So we have the two tables ready here under the results tab. So you can see the list of all the analysis runs you have done. So we will look into ischemic and the myogenic. So here you can open the results table and expand it. And then you can have a better view of things on the table, and then you can probably filter the table down to the specific rank thresholds. So here we want to have something very similar to, p-values. So we will bring it down to 0.06 and 0.05. And maybe we want to have a little bit more interaction here. So the table now has been filtered, but what if you're interested in a specific ligand? For example, over here, you can search for one that you have in mind. So this will highly depend on your field of study. But here, as just an example, we'll use call 1A1. And as you can see, the ligand complex here, all of these are from the fibroblasts, and then these are interacting with some of these myocardial parasites, and so on. So this is the, ischemic, so which makes sense that you have a lot of these sort of cell signaling that's related to inflammation, structural matrix, fibrosis, and so on. So what the fibroblasts are trying to do is they are trying to fix the cardiac tissue, and this is why you see a lot of these sort of structural matrix genes, and then these are the integrin complexes. And then here we will quickly go to the myogenic and see, like, with the same filtering here or similar filtering, like what is different with this. And here you can see that there isn't as much of those sort of communications between cells. That's because this is normal and there is no injury to the cardiac tissue, so there is no need for repair, and this is why you only see perhaps just fibroblast and parasite you only see one row of results here, after with sort of the same filtering that you apply. Of course, these tables you could export. You get a full table of results. Another feature we have, what if you want to sort of visualize some of these interactions, or perhaps maybe on a dot plot? All right, so let's take a quick look into the ligand receptor dot plot function that we have here. So this just allows you to quickly visualize ligand receptor pairs in specific source or target clusters. Of course, here, for example, if we looked at the results table earlier on, maybe we want to look at fibroblasts, and then, for example, here, neuronal and cardiomyocytes. Voila, we have a dot plot. For this, it's you can choose the top interactions. These are automatically added, but also you could just search for the ligand complex that you are interested in visualizing here. And then, for example, this one is for the ischemic sort of results. And if you want to compare with the myogenic, you can have sort of the same set of ligand receptor pairs here. And then you can go to the myogenic, plot the same clusters, and then plot the same ligand receptors, and then sort of have a visualized difference or comparison between these two results. And for here, as you can see in the legend, the specificity and the magnitude, you'll realize that it is different from the results table, because these are negative log 10 scaled values. So you will expect them to be a little bit different from the original table. So these are just some tools that will help you get started, and hope this was helpful.